So I should begin by welcoming everyone. Thank you for joining this Masterminds Masterclasses dialogue with young scientists. This session today is actually a pre-forum event leading to our inaugural Hong Kong Laureate Forum. So I'll begin by introducing myself. My name is Stephanie Ma, Associate Professor in the School of Biomedical Sciences, Faculty of Medicine at the University of Hong Kong. So I'll be acting as a moderator primarily today at this session. I am myself a cancer biologist and working at the university. We're very much honored to have Professor Ulrich Hartl with us as our guest host today. So just a brief background of Professor Hartl, of course. Professor Hartl is currently a professor and managing director of the Max Planck Institute of Biochemistry in Germany. Professor Hartl did his MD and Doctor of Med at the University of Heidelberg and his postdoctoral training at University of Munich in the lab of Walter Neupert and also UCLA in the lab of Bill Wickner. He also traveled the world and worked in various institutes, including Sloan Kettering Institute and Howard Hughes Medical Institute, before moving back to Germany in 1997. So Professor Hartl has been recognized, of course, with multiple awards and elected as a fellow of many prestigious academies and societies over the years. So this is really a long list. So I just pick and choose. So he is awarded the, of course, Lasker Award for Basic Medical Research in 2011, the Shaw Prize in Life Science and Medicine in 2012, and most recently the Breakthrough Prize in Life Sciences in 2020. So he is recognized for his pioneering work in the field of protein-mediated protein folding, and in particular for discovering functions of molecular chaperones like H-shock proteins in mediating protein folding and preventing protein aggregation. So the purpose of today's event is really to have our guest host share his research journey and personal experience with our younger generation of students. And today we're very happy to have all 10 of you. I see that we have some undergraduate students, some are currently doing their PhD students, and also a PhD graduate with us today. So what I'm going to do is to invite all our participants for the first round of discussions. So maybe one by one, what I'll do is to ask you to introduce yourself and also ask one question to Professor Hartl that you have prepared, and then we'll go on from there. So shall I first invite, I'll just go down the list then. Can I first invite Mr. Ka-Wah Kong, who is an undergraduate student at the University of Hong Kong, to begin.
Hi, Professor and everyone. So I'm Matthew and I'm currently a fifth year medical student at HKU. My question for Professor would be that, do you think that computer coding would be something essential for basic science research in the future? That means that even if I work in the lab, I still need to learn a bit about coding or those sorts of things.
Well, let me first welcome you all to this interesting round of discussion, and thank you, Stephanie, for the introduction. I look forward to having a stimulating exchange with you. Yeah, I was thinking about your question because some of the questions were submitted to me beforehand. I don't know much about coding, and I have people in the lab who can do some, but we have also specialists in the institute who do that when we needed it. For example, for certain bioinformatics analysis or things like that. Certainly, it's not a disadvantage, but I don't think it's an essential aspect for the kind of work that we are doing. Maybe it becomes more important in the future. But I wanted to make a comment more generally that many young scientists believe that the more methods they learn, the more methods experience they have, the more qualified they would be to do research, and more likely they would be successful in research. I think generally, if you have a really good background in relevant basic sciences, and particularly, of course, for the kind of work that we are doing, you would need to have good understanding of basic molecular cell biology, some biophysics and so on. Then you can really learn most of the things by doing, and the most important thing is to have defined your research problem and understand what are the methods that are required to address this problem, and that's what you would then usually learn in a laboratory where you do your PhD or postdoc, from people in that group or from your mentor, from your supervisor. So while it is certainly great to have experience with methods, various types of methods beforehand, there is always an opportunity to learn. And this perhaps is particularly important for people who come from, say, developing countries, students from developing countries who may not have such a broad opportunity to be acquainted with experimental methods. They oftentimes have a very good theoretical background, but maybe not have so much practical experience. So in essence, certainly a good idea to know how coding works and have some experience with it, but it may not be an essential aspect in my view. This depends, of course, which type of lab you want to join and what type of research they are doing. If you go to a bioinformatics lab, I certainly would say you must know coding.
Thank you very much.
Are you actually doing any research at the moment? You mentioned that you're a medical student, right? In the 5th year.
Actually, I'm now working as an intern in Professor Ivan Hong's lab. So I think we are currently working on the immune response of the COVID vaccines and some COVID-related projects going on.
Mm-hmm. That's interesting.
Okay, I'm sorry if I pronounced your name wrong. It's Mr. Gulas?
Yes, it's correct. Thank you. So hello everyone. First of all, I would like to thank you for the opportunity that you gave me to participate in this unique masterclass. So my name is Kyriakos Goulas. I'm from Greece. I graduated from medical school of Aristotle University, and now I do my master's degrees in precision medicine in clinical practice. So I would like to ask your opinion about personalized medicine. Do you believe that personalized medicine is the future of medical science? Also, are there any risks arises from this advanced type of medicine? Thank you.
Yeah, certainly I do believe that personalized medicine is where the future lies in medical treatment. And since you probably know more about it than I do, but it's very clear that it will become more and more practical to obtain large data sets on clinical and physiological biochemical parameters of patients in the future. Certainly, it will become feasible to have DNA sequence information, which will provide, of course, as you know, extensive information on certain risk genes, perhaps combinations of risk genes and so on. There will also probably be in the future the possibility to analyze patient samples, serum, perhaps other samples by high-resolution proteomics. In fact, I just read in preparation for this discussion, there was an interesting article in the ASBMB magazine, which is ASBMB is the American Society for Molecular Biology, Biochemistry and Molecular Biology, and they talked about the future of proteomics in clinical applications, whereby it is now possible to quantitate numerous proteins in different types of samples and first of all use this for diagnostic purposes, but also use it for following the progression of and the usefulness of certain therapies, and of course also in terms of personalized medicine, in terms of determining what kind of treatment a particular patient should probably get and what would be most successful. But there was also in this article mentioned that of course this type of information that will then become available has a potential downside to it in terms of the data privacy rules that would have to be followed, ethical concerns. For example, it was very interesting, while we all know that you can of course identify an individual based on his or her DNA sequence, now the proteomics methods have become so reliable that, in fact, I know this also from my colleague Matthias Mann at our institute, who is in fact one of the pioneers in this technology, also with regard to clinical development. These methods have become so accurate that you can actually, by combining different parameters and seeing the ratios, quantitative ratios of different proteins, you can also perhaps identify an individual. So we will have to be careful in how we treat this kind of data in order for it not to be used by certain organizations, perhaps the employer, to make certain predictions about what is going to happen with this particular individual in the future and so on, insurance companies and all these matters. But I think it is very clear that this type of analysis will greatly facilitate individual treatment of people based on their actual makeup. There was also an interesting example whereby a researcher who himself was involved in carrying out this type of proteomics analysis for many years, in fact, apparently from childhood, suffered from high cholesterol levels and was told that he should try to do exercise, lose weight, have a certain diet to get this under control. And he, in fact, refused to take any pharmacological treatment. But then when he carried out his own proteomic analysis, where he was supposed to be a control individual, he discovered that he, in fact, has a particular lipoprotein risk factor of ApoE allele, which indicated that there was no chance for him to ever get this under control by diet or exercise. And from then on, he started to take statins. And in fact, he found that in his family, that was quite common, the same problem. So now they are all taking statins and seem to be in a much better state. Now, this is a very simple example because I personally think this guy should have used statins right away, since these are, in my understanding, fairly safe drugs. But of course, it may be a difference whether you take it at the age of 60 or in your 20s. In any case, there is also possible benefits. And if this data would have been completely anonymized, as it usually would be in such studies, it would have been impossible to go back and find out, who can we help by understanding this now and by identifying this particular problem. So I think there will be tremendous progress, and I guess in certain areas, cancer therapy, I think Stephanie knows more about this. Personalized medicine is already being very much in use.
Yes, most definitely. For a lot of, in particular, I guess like breast cancer, I mean, that does, there's very like HER2, and it's been shown very effective. So precision medicine for cancer therapy, for some type of cancers, is very effective. And I think that is really the way forward for the field. Yeah. By the way, I want to emphasize that, for those that, you can ask, feel free to ask follow-up questions to what,
Of course, you should all feel free to interrupt anytime. And if you have any remarks to the question that was just being asked or anything else, then we should be able to talk about it.
Yeah, so don't be shy. Just unmute yourself and just jump in.
I mean, since Stephanie just mentioned Herceptin, the HER2 gene and all that, and Herceptin was absolutely a breakthrough in breast cancer therapy. And I mention it because Axel Ulrich, who was one of the developers of Herceptin, is a colleague or was a colleague, is now retired at our institute. So tremendous progress has really been made. And this becomes much more specific in the future, I would imagine, adjusted to the specific individual.
So Emma, can you try to end your microphone again?
Yes, can you hear me now?
Yes. Yeah, we can hear you, Emma.
Oh, thank you. Hello everyone, I'm Emma and I'm a year 3 bioinformatics student from University of Macau. And my question is it possible to cure all the genetic disease? Thank you.
Well, actually, nothing always comes in completion, I guess, and certainly not in the life sciences. I'm not an expert in that area, but I believe that there will be very substantial progress in the future in curing genetic diseases. When this will happen, I'm not so sure. Of course, people are working very hard to develop CRISPR gene editing for this purpose, but also other technologies. I guess it will depend a lot on, that you have access to the type of cell type and tissue type where the problem occurs. At what stage of development basically can this type of therapy be performed? I'm optimistic that some of the genetic diseases, but the monogenetic diseases obviously can perhaps be cured, but it will be a lot more difficult for diseases that have a more complex inheritance patterns, which include some of the major diseases that our civilization, humanity has to find ways of treating, especially also if you think about neurodegenerative diseases, many diseases that occur in an age-dependent manner. I'm not so sure that we will be able to use these new technologies a lot or very productively in these cases. Stephanie, perhaps you can say something because you know more probably about, you used CRISPR screens in your own work, did you not?
I do.
Yeah.
But of course, just on just screening, I think to initiate a project to identify, things that may be critical for, driving cancer, right? Of course, everyone knows about the case where, there was a person that tried to, use CRISPR editing to make, and got jailed for it, right? And it's just, it's also interesting to explore into ethical issues about these things, right? Of course, using, if you have views on this, whether, I guess, when it's safe to, use CRISPR editing in which circumstance, and whether, we can actually adapt this technology. Emma,
there is still not complete clarity, I think, about possible off-target effects and so on, whether we can improve the specificity to a level where this cannot happen. But on the other hand, I think it's almost unavoidable that it will be used for some purposes. And once that has started, the development might perhaps be even more rapid than we think now. Ethical issues are tremendous, obviously. Yeah.
Anyone working on CRISPR here? CRISPR editing here?
We only use this in the lab as a tool, which of course is absolutely fantastic. This has tremendously expanded things we can do, the kind of experiments, and also made accessible types of model organisms that were not easily manipulable genetically, but can now be done. So in that sense, of course, this is already, has obviously deserved all the praise and awards that were given for this type of development.
Okay, someone asked, at what age should we then start with genetic editing? This is in the chat room.
At what age
should we then start with genetic editing?
Well, as I said, I'm not so sure about how this will go, but it depends on the particular problem. This might vary a lot dependent on the type of genetic problem that has to be repaired.
Right, let's go to Nathan. Can we invite Nathan from University of San Carlos?
Yes, good evening. Good evening, Professor Hartel. I am Nathaniel Lior. I am from the University of San Carlos here in the Philippines. And I would like to ask something more personal in your career as a scientist. What was the biggest challenge you have experienced in your career as early as a young scientist? Thank you.
Okay. Well, actually, I thought about this quite a bit, and first of all, I would have to say that with hindsight and retrospectively, I have been extremely lucky in many ways that good things and combinations of good things happened at the right time, and I was at the right place at the right time. So I think it's very important to realize that for a successful academic career, many factors have to play together, and there are elements that are not really predictable. However, I would say that the major challenge was to really identify the kind of research field in which I could make my mark and in which I could do so independently on my own. And obviously this is a very important transition in every academic career, from, say, at the level of postdoc to becoming then the person who actually drives his or her own research, has ideas, and is able to carry out the work as a principal investigator, which means that you are responsible for the type of research problem that you have chosen, how you address it, and you are also responsible then for the results that you eventually publish. In my case, this was possible because the research problem developed, and as it probably is the case for most scientists, it developed from my postdoctoral work. I was at the time working, as Stephanie has mentioned before, I was working with one of the most famous German cell biologists at the time, Walter Neupert in Munich. Walter Neupert had pioneered the science of the research and the understanding of how proteins are sorted within cells into particular compartments, and he studied the mitochondria. So in his laboratory, we studied how mitochondria, as you know, are the organelles in our eukaryotic and mammalian cells that required for energy metabolism and many other functions, how these organelles import their proteins from the cytosol after they are made in the cytosol on the ribosomes. At the time, it became clear, and I had contributed to that also in some experiments, but mostly from prior work in Neupert's lab and in the lab of Gottfried Schatz, it had become clear that protein chains traverse the mitochondrial membranes in an unfolded state. And so we realized that we could use this particular system to study how the protein folding reaction would then happen after the transport of a protein into the mitochondria, after its translocation across the membranes. I was also very lucky at that time to have a very productive collaboration with a colleague in the US, Arthur Horwich, with whom I shared many awards, in fact, many years later. And we could use this model system to address a very fundamental problem that people had realized to be a fundamental problem. And what we discovered was that this protein folding reaction was not just occurring on its own spontaneously, as had been suggested, but it required a particular machinery, a molecular chaperone. And this then put me on a track of investigating this reaction, of this process of assisted protein folding. And I then made one, I think, very critical decision based on what was generally known about the type of proteins that were involved in this process as chaperones. It was clear from our work that apparently these are stress proteins in many cases. There had been prior descriptions and analysis of stress proteins, in particular with regard to their inducibility under various conditions, such as heat stress. And the best work, the easiest system at this point was E. coli. So I decided to leave the mitochondria behind and start to investigate these reactions in bacterial cells, which after all are very similar to mitochondria. And at that point, I could make very rapid progress, which was, first of all, very important. But another important aspect, which I didn't really think about at the time, but it became clear to me later, that my mentor, Walter Neupert, was not interested in bacteria. And at this point, he, of course, continued his successful research on the mitochondria. And I had this whole area for myself and could work on it as an independent researcher. I then also accepted a position in the US, which provided me with complete independence very rapidly, rather than perhaps having to wait for a few more years until I would have been able to acquire such a position in Germany. Also, the position in the US did not require me to spend a lot of time on teaching, which turned out to be a very lucky circumstance. So I could focus on the research, and I had very good funding. So many factors came together, but I think identifying your own problem and acquiring scientific independence, these are the major challenges that everyone will face who wants to become a successful scientist, especially in basic research. And once you've done that, then of course you still have to continue doing good work and be lucky, but that's probably a major energy barrier, so to say, that you have to overcome.
I can't agree more with what Professor Hato said. I remember when many years ago, when I interviewed for my faculty position, what I got asked was two questions. The first is, how will you differentiate yourself from your current postdoc mentor? And the second is, 10 years down the road, when you're up for a promotion for a professor, say, for example, what do you want to be known for? I think these are the hardest questions. I mean, still today, I still ask myself, right? Because, what do you want to be known for in the field? I mean, okay, we think of, okay, Stephanie, then, then people will, automatically associate you with some kind of science or topic. Then I think you'll be sort of successful in that sense, right? But, today, I still ask myself this question. I don't, and I think what Professor Hato mentioned is, I think for all scientists, this is the challenging question.
But sometimes it helps to step back a little bit and try to look at what you have done and what you are doing from the outside. Of course, it is, I think you have to realize one thing. There are thousands of outstanding scientists, but not everybody has the good fortune to make a major discovery that changes a field or opens up a new field. So to be lucky, and of course, to be lucky alone is not sufficient. You also have to then have to make use of it, of a situation. I think I would have to say that I was very lucky, but I was at least able to recognize what the opportunity was that offered itself. Yeah. So that's what you have to look for. And of course, the chances of making this step successfully are much greater if you are already in a laboratory where frontier science is being done, where people are really discovering things and driving a field forward, which was absolutely the case in Walter Neupert's lab. And again, as I said, I was just really lucky to be accepted to that lab. It had not been planned. It was just a result of the fact that my doctoral thesis was sent from Heidelberg University to Walter Neupert in Munich for evaluation. And as a result of this, he invited me to come to visit his lab to talk about my work and learn about what they were doing. And then he offered me a postdoctoral position. Otherwise, I think if this would not have happened, I would certainly not be sitting here and talking to you today. Okay, so go to a lab that is doing great work, if possible. Then you have a much bigger chance to find your own niche, your own problem that you can derive from whatever is being done in that setting.
Okay, so can I invite Ying Hon from the Hong Kong Polytechnic University, please?
Yes, I'm Yinghong Si from the Hong Kong Polytechnic University, and I am a last year PhD student in the School of Optometry. And I guess I'm gonna ask relatively spiritual questions, like how often you fail until you reach the success nowadays.
Yeah. Well, I think failure is a very important and frequent part of doing science, because when we do an experiment, we do it because we don't know what the outcome is. And unfortunately, an experiment can have a negative result, which is often seen as a failure, even though it is of course an important step in the whole process. Oftentimes, ideas that we have, how something might work, simply don't pan out and fall apart. Things can turn out to be simply too difficult, are not practical, and so on. So you have to have a lot of frustration tolerance, but hopefully at some stage in your career, you will actually find something out. And this doesn't have to be an earth-shattering discovery, but it should be something where you feel that you have now seen something that maybe no other person, no other scientist has observed in that way. That would be fantastic, obviously. And it shows you that you have been able to do that. And once that has happened, I think it becomes a lot easier to take other frustrations, to cope with other frustrations, and still be excited. There were other questions that were submitted. What is the kind of property that a scientist must have and so on in order to be successful? I think one of the most important things is that you must have the excitement for the work. You must feel that this is the best thing that you can spend your time with. Occasionally, what you do is going to work, and that helps you to overcome other frustrations. Also, one has to say, of course, as soon as you become a principal investigator and you have a group of scientists working with you, students and postdocs, it'll get a lot easier in terms of frustration, because hopefully there is always someone in your group whose experiments are actually working. That's what gives you satisfaction and helps you to take care of the others whose experiments may not be working yet at this point, or have to be restructured and so on. Of course, the frustration is the greatest when you are the only person at the bench working on your experiment, your PhD, your postdoc experiment. And of course, you know that if you want to be successful in science, you have to get some results that can be published in a good journal, because that's what will then be the basis for your next career step and so on. That's when the frustration pressure is very high, and later maybe it subsides a bit, but it will be replaced by other pressures. It will be replaced by the pressure to be successful with your grant applications, for example. Because if that doesn't work out, you will not be able to continue your research. You will have pressure to be tenured, to be promoted to a tenured position, and so on. So it's not ending very soon, but after all, you will have lots and lots of experience, and that helps a lot in the life sciences to cope with these problems.
A lot of times what you see as a failure, you can always learn from that failure. You always, there's always something that you can learn, although you seem, that experiment or that setting may seem to be a failure. So I think being optimistic is also a very important.
I think what Stephanie says is very important. I have seen many in my many years now as heading a lab, have seen many different types of personalities of co-workers, and certainly having a positive outlook on things can be very helpful. And also, I think to be communicating with others well, to discuss your problems with others helps you to learn from, as Stephanie said, learn from a failure and obviously avoid it in the future, or plan your experiment better so that it will give you a result that forms the basis for the next experiment. That's all we can expect from an experiment, that it will give us a clear result that tells us how to proceed.
All right, next, can I invite Diana from the Chinese University of Hong Kong?
Hi,
Hi,
it's
Diana.
such an honor to speak with you. So I'm currently a clinical lecturer and a PhD candidate with CUHK. With such a distinguished and long career, I guess I have more of a personal question too. And I'm wondering what still inspires you in this difficult journey of scientists, like discovery and like filled with failures. So
this is a very interesting point, and in fact, sometimes I'm working very closely with my wife, Banerjee, who is also a very accomplished biochemist. Sometimes she asks me also, why do you always want to continue? Why do you always want more? I think it's once you have felt this excitement of understanding something, of discovering something, you will simply get hooked, and it's like an addiction in a way. So you cannot simply stop doing that. And this is in fact one of the biggest problems that scientists at some point is also facing, that it's not continuing forever. Especially in the European system, we have a retirement age. We can get extended, and of course everybody is trying to continue as long as possible. But at some point, I will no longer be the director of a department, and it's a very hard thing to cope with that idea. But in the meantime, I am just excited to find out what my students and postdocs have done, what results have they obtained in their experiments, and we are making slow progress in certain areas, and that's extremely satisfying. Another thing that I think provides a lot of motivation is that the ability to interact with young people like yourself and the rest of the group, and of course the daily interactions with my students and postdocs is just a wonderful experience. It's a real privilege to do that. I think it also helps a scientist to stay young, maybe not physically, but at least mentally. You are interacting with people who have a very different framework, the way they see the world and what's important to them. But we are all united in this passion for doing science. And I have just at the moment, again, always had, but certainly at this time, a really fantastic group of students and postdocs who are interacting very productively, who help each other, who contribute different expertises. And I have to say, I learn a lot from them. That's really fantastic. Now, more generally, while we have, of course, learned, that the progress in the molecular sciences has been tremendous. But if you think about this, how long does this really go back? Then you could say maybe 60, 70 years or something, not too much longer. And there is a whole universe still of how the cell functions that we don't understand. So I don't think we are running out of fundamental problems that can be studied. And as you know, the new problems become accessible for analysis as new technologies develop. We have had in recent years very important new technologies developing in different areas of biology. So lots of new opportunities, I think, will arise. I think the cell is just such a wonderful system where we have now a basic understanding of some of its parts, but how it all plays together and forms a functioning module unit is, at least to me, still is really a wonder. And we cannot obviously take a cell apart and put it back together. Even if we were able to put the molecules back together in the same quantity, as soon as we disrupt a cell, it will no longer be possible. So there is still something that we really don't understand. And this is going to happen in the next decades, I assume, that people will make tremendous progress. So to me, this is the equivalent to space science. It's much more accessible, obviously. But in the microscopic world, we have this tremendous universe of cellular functions.
So what do you think is still
us,
a mystery to you for the cellular functions?
Of course, I mean, we have our own research problems, which are very much related also to a group of diseases caused by protein misfolding and aggregation, particularly neurodegenerative diseases. We don't understand how cells really maintain a functional proteome. We don't understand the network of factors that participate and how it reacts to various types of influences, how it is regulated, how it is programmed during the aging process. We understand now that our proteomes somehow lose integrity, begin to deteriorate as certain cell types age, in particular neuronal cells, which cannot regenerate as well as other cell types. We don't know why this happens. We know that as a consequence of it, proteins misfold and aggregate, and this is an important cause of neurodegeneration and dementia. So we really would like to understand how we can prevent this deterioration or delay, shall we say, so that we could perhaps extend the healthy lifespan of humans and most importantly, postpone the occurrence of neurodegeneration and cognitive decline, which I personally find the most threatening and brutal thing that can happen to a person. I mean, cancer, of course, is terrible, but to lose your memory and your personality is just really extremely cruel, not only for yourself, but of course for your family, for your loved ones, and for the whole society, given the amazing cost that is generated for the care of these people. And we have no treatments at all at this moment that would really work. So these are obviously things that we would like to address. But then also, as I said, while we know how DNA replication works, we know how transcription, translation, folding perhaps even works, we know about many signaling pathways, but how all this is integrated into a functional system, I don't think we are close to understanding it. And then, of course, apart from all that, the biggest problem obviously is how does the brain work? How does thinking work? What happens when we speak, when we formulate a sentence, when we make a calculation? I don't think anyone has even the slightest idea really how this works.
Yeah, it's true.
Lots of things that still needs to be unraveled, of course.
Yeah.
Never ending.
So it's not that always people who know that people in your own field who say this field has now reached its end, or this is now saturated, or people have said there is nothing new to be discovered in physics anymore. And then suddenly major things are being discovered that nobody even expected, which, by the way, of course, is one of the most important criteria for what a discovery is, that it is unexpected, and it happens all the time. So I'm sure that even in our area where we believe to have fairly good understanding, there could still be major surprises.
So we'll move on to the next person, Fernando from Brazil, please. Thank you.
Hello, it's a pleasure to meet you all. Well, I was once told that in order to be a good scientist, you have to have an idea that someone wants to buy and someone is willing to pay for it. This means you need financial support to do good science. If we look in Wikipedia for the Shaw Laureates, mainly come from United States, United Kingdom, Germany, places where they're having good financial support. Yeah. Where I come, we don't have good financial support for science. So my question is it possible to do science with little resources, and how to do it?
Yeah. So I thought a lot about your question. First of all, what your opening statement, I think maybe we can pick up on that. You said in order to be a successful scientist, you have an idea that somebody wants to buy. This brings me to a very fundamental point, namely what kind of science, what kind of research should be supported. We have two major areas. We have basic research and we have applied research. Now, in the Western countries, industrialized countries, in many of them, there has been a long history and a strong culture to support basic research. And by basic research, I mean essentially investigator-driven, curiosity-driven research that has no obvious application, at least at the time when it is being conducted. It could be completely without any possible application that anyone could think about at that point. But it will generate knowledge. It will add to the knowledge base of humanity, and that is being recognized as a cultural value in itself and is being supported by a particular society in a particular country or region. That means they are using essentially taxpayers' money to fund this type of research. This has been probably one of the most successful endeavors that humans have ever invested in, because since we are talking about the life sciences, I would say the vast majority of real advances in medicine have resulted from this kind of curiosity-driven research that was performed at a time without the intention necessarily to solve a particular medical problem. And there are numerous examples. Just take the present development of the COVID vaccines, which became possible, the mRNA vaccines, because somebody had tried to find out why do we get an immune response when we inject an mRNA molecule, and they found that mRNAs can be modified, or other RNAs that didn't cause the problem were actually modified. So tremendous progress in basic understanding has helped then also to make very rapid advances in developing these vaccines. For example, I don't think this would have been possible 10 years ago. And we can all be proud as scientists to have contributed overall to this body of knowledge that helped with that. Now, obviously, as you mentioned, this type of resources are not available in some countries that are, still fighting with a lot of other problems, and they cannot afford to use taxpayers' money at that extent that US or UK or Germany or the European Union can to fund basic research. So what can you do in such a country to still make important contributions? Obviously, you could perhaps I have no problem with translational research, by the way, and I think it's a very important area to be supported as well. And of course, it is more typically supported by companies, by industry, by pharma companies, biotech companies that want to make a certain development in order to also earn money, which there is nothing wrong with that, because it only works if it actually helps people, at least in the long run. So I'm pretty sure that in Brazil you have such opportunities perhaps as well, but they might be limited. But I was also thinking, what could a country of Brazil offer you in terms of resources that other countries do not have. And of course, what comes to mind, but I'm not a specialist obviously here, is the enormous natural resources that a country like Brazil has in terms of biodiversity, in terms of natural compounds perhaps that could be researched or identified in those diverse organisms. So I could imagine that there might be areas of quite basic research that you would be potentially in a position to do very well. And then of course there is always the option, which many scientists from your parts of the world, Argentina and others, have followed, is to leave your country at least for some time and go to the US or another country and be a postdoc and perhaps become a professor. It's not uncommon, as you probably know. I know several South American scientists, but almost none of them are actually working in South America or Africa or for that matter, and it's quite common to do that. But of course, you might not want to leave your home country for many reasons, for many good reasons. So obviously you have a problem, and there is no simple solution. But I think at least perhaps for an educational period during your career, it could be advisable to go to another country where you can then participate in the wealth that they have and the resources in terms of supporting science, especially if it's an experimental science. You need resources, otherwise there is no way to be competitive. For example, if you are a structural biologist nowadays and you do not have access to high-end electron microscopy, you can just as well forget being a structural biologist. It's not possible, right? So in Brazil, there might be, I don't know how many universities there are that have this type of equipment. I'm sure there are some, but a lot fewer probably, especially given the size of the country, than you would find in Western Europe or in the US. Or in Hong Kong for that matter, and in China, of course.
It's very useful.
By the way, this is just something that I was thinking spontaneously, because I think China has, and Hong Kong and China have a tremendous potential and already doing unbelievably productive and important research, but also with a really major support in terms of resources. And I don't think that Hong Kong and China are necessarily the countries where a postdoc from Brazil or Argentina might want to go, but maybe it would be a good choice.
Yeah, I mean, Hong Kong and China, we're very fortunate that the government is putting a lot of emphasis and a lot of financial resources into research and innovation in the past 5 to 10 years. I mean,
exactly.
I came back to Hong Kong in SARS in 2003, and the landscape is completely different in terms of research funding from then to now. And we also have schemes that actually support overseas students, from anywhere in the country that probably do well academically to come abroad and to come over to Hong Kong, and we give you scholarship for your PhD or for your postdoc. Definitely a lot of, I'm sure in the US as well, right? So I think good opportunities.
This has always been a really successful investment. For example, after the war, of course, German science was, pretty much destroyed for many reasons. But then it was built up again, and there was the realization that we had to pay young scientists to go abroad and learn new developments, especially at that time in the US. And in the hope that a fraction of those would come back and would then fertilize the system and bring it back to a higher level. And I think in the long run, this worked very well. Maybe in the beginning, many, particularly the more successful ones, remained abroad for a long time, but later many of them came back. and this was a very good investment, to pay them a fellowship without strings attached. And I think this is also something that China has been very successful with that. And now, of course, they offer fantastic positions. I've had a number of Chinese excellent postdocs from China and students who have been offered really great opportunities in their own country to do research.
In the interest of time, let's move on because we still have 3 applicants that haven't had a chance
continue
to.
Then.
Yeah. So I apologize if I pronounced your name wrong. Is it this?
It's called.
I was wondering about it.
Sorry.
Okay. You are from Leiden, from the Netherlands, right?
Yeah, thank you very much. I'm a 5th year medical student, but I've stopped my studies now, and I'm currently a 2nd year PhD candidate in Leiden University in the Netherlands. And actually, my question we already touched upon. So I was thinking of another question. How has international and also national collaboration affected and maybe propelled your research to where it is now?
Okay. Yeah, it's good that you bring that up, the aspect of collaboration, because there is very strong support and incentives given by various funding organizations, in particular also the European Union, the ERC, to support collaborative research. And I have been myself involved in various consortia in the European Union, for example, where members from various countries participated, and it was very successful. I would say, while I definitely support these kinds of initiatives and have been engaged with them and benefited from them, I do not think that personally for my own research career it was absolutely essential or critical to have these types of collaborations. And by that I now mean those collaborations that are formalized in a certain funding instrument where you are basically forced to collaborate, okay? But I believe that scientists, and since science is an international endeavor, will always collaborate and find collaborators to solve particular problems. And that's what I have also done in my own career. But independent of this type of organized consortia, because we are exchanging information with other scientists at meetings, we are finding out about research interests other scientists have, and we get to know each other, and then collaborations can arise very naturally where they are most fruitful because the partners contribute different aspects. I do not believe that collaboration per se and in itself is a value that should be, for example, used as a criteria for evaluating a scientist. Has this scientist collaborated, or has he not collaborated? Has he been collaborative, or has she been collaborative? Sometimes you are now hearing this in committees, and I don't think that that's a good question to ask. What should be asked is, has this scientist discovered something? Has he or she contributed? Has he or she advanced the field? And in many cases, of course, real advancements do involve collaborations, but it's not a necessary thing that provides value in itself. At least that's my personal belief. But I strongly believe in the value of collaboration and in openness and so on. And exchange, that is very clear. But in my own work, I would say collaborations have been important, but we probably would have reached our goals with more or less collaboration as well. I mean, it wasn't the most critical aspect.
Thank you very much, and also thanks for the organization.
I mean, I think I agree, but I guess I do agree to some extent. Like forced forced collaborations are not useful. So probably just to bring in the money, but it's probably not the most fruitful. But I guess with a lot of interdisciplinary research going on, I think for me at least, engaging with different collaborators that are not in their field will actually help you open your mind to different things. Because one person definitely cannot know everything, right? And when you collaborate with someone that is completely, say for example, I'm a cancer biologist, and I want to extend into cancer immunology, but so I'm currently collaborating with a immunologist, a basic immunologist, which probably looks at the same question from a different angle. And I'm learning a lot from my collaborator in that sense. And I think that will help me. It has helped me open up my my perspective a little bit in things. So I guess collaborations are may not be a must, but it's also useful.
I completely agree with Stephanie on this. And maybe this didn't come over exactly as I wanted. All I'm saying is that the instrument of forced collaboration, I don't think that's really working so well. But of course, given the interdisciplinary nature of science, you cannot have expertise in everything. And it is extremely useful to combine expertises from different collaborators. In fact, I have done so, and oftentimes, and I've done so in the very beginning of our work. I mentioned before the collaboration with Art Horwich, and he was a geneticist and I was a biochemist. So that was already a good example for how different expertises can come together and synergize.
Okay, so Sima, would you have thank you for your patience. I think she's trying. Oh, there we go. Oh,
yeah, now can you hear me?
Yes, we can.
Good evening, everyone. And Professor Hartel, this is such an honor to interacting with you in this session, So, my name is Sima Banerjee. I am currently a postdoctoral research fellow at the Department of Ophthalmology in Johns Hopkins. I am a 2020 graduate from the Hong Kong Polytechnic University. So I have completed my PhD and I have just joined Johns Hopkins. Recently, we had a session and somebody told me that, science is exciting as well as it is a roller coaster between, mixed emotions, between achievements and rejections. So you have a lot of rejections as well, your grants are rejected, your proposals are rejected. So it is very important to find that balance and being calm into this career, and as you had an exciting research career. So my question to you, what are the 3 tips you would like to give us, all the young researchers, to become a good researcher, in the field of science?
Yes, I think I can only give you 1 tip. And the answer is actually there are no 3 tips. It's very difficult to make such general statements, I would say. It is absolutely true what you said. There are constant rejections, and this does not end. I still have, we submit a paper to a journal and it may or may not be accepted. Of course, and if it's rejected, the disappointment is still the same as it used to be when I was much younger. So this doesn't really change a lot, and when it is accepted, we are still as happy as we used to be. So this, but there could be one consequence that I think would be wrong. Perhaps I could tell myself, why do I still expose myself to this type of possible rejection? Why don't I just submit my work to a simpler journal where it will most likely be accepted? Or why do I even submit something at all? And that would be the biggest mistake, of course. So we always have to expose ourselves to the criticism of our peers. With regard to what kind of advice, obviously we talked about the ability to take frustrations. And I think this can only be done if you really feel a deep excitement and passion for for doing research. If you wake up in the morning and think there could be nothing better than going to the lab as soon as possible and continue your experiment or evaluate something that you have done the day before. If you feel that, then you will be able to take frustrations a lot easier. But also, if you feel that, then most likely if you have talent to do this work, which of course is also important, but I assume that you all do, because otherwise you wouldn't be here. You would have to have had already during your high school days, you would have had strong interest in science and all that. So I assume that all that is the case. If you feel the excitement, then you will also have success. You will do something that will work, and that helps you become even more excited and continue do the hard work. Obviously, if you want to have financial success in life, then choosing this career may not be your best path, even though there have been some scientists who have been financially successful as well, but I think it's a strong minority. So you will be able to have a good living if you are successful as a scientist, but you will put in a lot of hours, and you have to be completely self-motivated. Nobody at some stage, nobody tells you anymore what you have to do. It has to come from yourself, from within yourself. You don't really have a boss in the classical sense. You may have a reviewing committee, you may have an advisory body that comes every couple of years, and this may or may not have consequences, but generally it all has to come from yourself, and that's of course very different. That's a privilege on the one hand, but it can also be a problem. And we already talked also about the choice of lab in which you want to have your different career stages. Obviously, if you are a successful scientist and you look where did these people come from, then you usually see that they already have been trained in other highly successful labs in the majority of cases. They have been at very good places in the beginning. And so if you can get that will make it much more likely that you are also advancing. But there are many factors, as I said repeatedly, and it's hard to give a general recipe. Unpredictable circumstances play an important role in science in such a career, as they do probably in most other careers as well, but perhaps even more so in academic science, which has so many facets and factors that influence it.
Thank you.
So last but not least, can I invite Alexei from Russia?
Yeah. Hello, everyone. Thank you for inviting me, and it's an honor to meet you, Dr. Hartel. Maybe you partially asked my question, but still, my question is, what are now the most challenging questions in molecular biology from your perspective?
Yeah, actually, I think we talked already a little bit about that. And from a cell biochemist perspective, I would say that understanding how fundamental cellular pathways are integrated into a system is not yet understood. And certainly we are far from being able to build a cell from scratch, basically from its constituent parts. And one of the perhaps most famous statements that have ever been made by a famous scientist, Richard Feynman, was, "What I cannot take apart and put back together again, I cannot understand." So in that sense, we still don't really understand how a living system functions. We don't yet know what are the minimal components that we have to put together to get a self-replicating living cell or even organism. That is still, I think, in the distant future. It's an area that is now being described as synthetic biology, which is making progress, and more and more people are interested in that. Certainly there are possible applications from this. Another area, of course, in our own research interest, I would say perhaps the most interesting aspect is to understand why do we age? What is really the reason why cells decline, certain cellular functions decline? Most prominently, the ability of cells to maintain their proteomes, to fold proteins properly and maintain their structure. That is clearly a major function that plays into almost everything else, which undergoes decline in an age-dependent manner, and which then eventually facilitates the manifestations of numerous diseases, age-dependent diseases, not only neurodegeneration, but also others. I would really like to understand better what are the major limiting factors in that process, and of course, perhaps then also find ways to kind of slow down this deterioration, not necessarily to extend the absolute lifespan of humans, which again would be ethically probably quite critical, but at least delay the onset of some of these diseases that are strongly reducing quality of life. So being healthy for a longer time, I think would be fantastic, and it would also, of course, reduce the financial burden that is posed by some of these age-dependent diseases. In particular, if you think of Alzheimer's alone, the amount of resources that simply have to be used to take care of these patients is tremendous. Another area of research that I know about, because my collaborator and wife Manerjee Hartle is working on it, has to do with the fundamental question: how can we feed the growing population? How can we produce enough food source to support an ever-growing population, in particular in the face of climate change that is reducing agricultural production in many parts of the world, mostly those parts of the world that are not responsible for the climate change. And this is related to our research because one of the fundamental proteins that is required to produce biomass through the process of photosynthesis is a protein called rubisco, which is the enzyme that absorbs CO2 from the atmosphere and builds biological molecules, sugar molecules from it. And that is responsible for the growth of all plants and indirectly for the growth of all animals, directly or indirectly. And we need to understand how we can improve the performance of this enzyme and of related pathways in photosynthesis to increase the yield of crop plants. The reason why we are interested in this protein is it is perhaps the most chaperone dependent of all major proteins. It's the most abundant protein in the biosphere, and it is dependent, as she has discovered, as Manerjee has discovered, on multiple chaperones that are required not only for the folding of this protein, but also for its assembly into a large oligomeric structure, into a large protein complexes, multi-subunit complexes. And it is also, and chaperones are furthermore required to keep this protein in an active state. Otherwise, it undergoes self-inhibition. It needs to constantly be repaired. And there's a whole machinery that cells are building to supply sufficient CO2 to this enzyme. Again, another way that can be understood and manipulated in terms of improving its performance. So it's a fantastic example how our basic research might eventually benefit agricultural productivity in the long run by finding ways to make this protein better, since we now know how to make it recombinantly. This has not been possible until recently. We can now express this protein in E. coli cells by putting all the chaperones from plant cells into the E. coli cell. And now we can make mutants and can select potentially better versions of this enzyme. So an application that was completely not considered at the time when we started working on these chaperones. The fundamental chaperone is the same one that we already discovered in the late 1980s when all this field started. So I think there are enormous opportunities in life sciences, hopefully for many of you. I can say this, perhaps because anyway we are reaching probably the end of our discussion. I hope that many of you will have access to working conditions that allow you to fulfill your dream in investigating these problems. The time when you do so, then I think all that counts is that it's an exciting problem, and sooner or later there is a good chance that it might come to benefit in medicine or in some other area.
Thank you, Professor Hartel. So I've just been signaled to wrap up the discussion because we have the hour and a half have gone by really quickly, and
that's
as always, good times always pass by more quickly than we wish it will. Can I invite Professor Hartel maybe just to end the discussion by just sharing a few words of wisdom, to end this. I don't know.
I try to share so many words of wisdom. With my limited wisdom. But as I just said, I want to really make a strong point in supporting the value of basic research. I can say that I've been very lucky to be supported by the Max Planck Society in Germany, which is a research institution, organization that devotes itself to basic research. There are other organizations in the UK, MRC, and France and other countries that have similar organizations, certainly also the US. And I think there is very important value in supporting this type of curiosity-driven research. We should not demand from researchers that they only work on an applied problem. What is the use of something? This question. Politicians oftentimes want to see the use. They have problems supporting basic research and giving researchers freedom. They want to have control what happens with the tax money that we give researchers, but that is a mistake. It's short-sighted because the future comes from the discoveries in basic research. So I hope that at least some of you will be able to either do basic research themselves or at least work on the results of basic research and develop them further for applications in medicine and in biotechnology. Our civilization, our world faces a lot of problems that need more research rather than less, and need more investment in research rather than less. And I hope that this is happening and that many of us, including the younger generation, yourself, will benefit from that. Thank you a lot for your questions and for bringing up some of the most interesting problems. Obviously, we have no solutions for many of them, but thinking about them is already helping. I have to say, I always found this type of discussion very motivating, inspiring, and it helps me also to again realize how great it is to be a scientist and what a privilege it is.
Thank you everyone for your participation today. In particular, again, thank you, Professor Hartel, for your insightful and inspiring sharing with us. And I myself also learned a lot from you, and also from all these young people and your questions. Okay, so this brings us to an end. Thank you very much again.
Thank you, Stephanie. All the best, and I hope to meet in the future.